Well, welcome everyone to the Center for Great Lakes Literacy Students Ask Scientists presentation. My name is Ginny Carlton. I'm the Education Outreach Specialist with Wisconsin Sea Grant, which is hosting today's Citizen Science presentation. Um, Wisconsin Sea Grant is part of the Center for Great Lakes Literacy, which is a coalition of educators based across the Great Lakes Basin. And we do have representatives from the various states who have signed up for today's presentation. Today's Students Ask Scientists presentation will be with Ben Peterson from the School of Freshwater Sciences at the University of Milwaukee. And before we get started with that, I also want to let you know that besides myself, we have a colleague named Ann Moser, who's our Senior Special Librarian and Education Coordinator here with Wisconsin Sea Grant, and she'll be helping to manage the question and answer series as well as some of the polls. Please do feel free to write questions into the question and answer field as Ben is doing his presentation so that you don't forget about what you want to ask. If there's something Ann and I can answer during the presentation, we will go ahead and do that. Otherwise, we'll save the questions to the end. And with that, I'm going to stop sharing. I'm going to ask Ben to go ahead and set up his screen share and ask Ann to launch the poll. The poll is going to ask you what state you are located within, what city, if you could please type that into the response area, your zip code, how many individuals are joining you today in the room with you, and whether or not you have, if you're an educator, we'd love to have your name. This helps us with our own reporting requirements to the Center for Great Lakes Literacy and to NOAA to be able to continue to bring you these types of presentations in the future. I'm going to ask Ann to keep the poll open for just a little bit longer so people can respond as they're coming into the event. We still have attendees coming in. And so, Ben, I'm just going to ask you to hold off for a couple moments before we get started with that. And Ann, you can decide when you think folks are in and you want to close the poll, and then we'll go ahead and turn it over to Ben. Thank you, everyone, for joining us. Again, if you're just coming in, my name is Ginny Carlton. I'm the Education Coordinator. I'm the Education Outreach Specialist with Wisconsin Sea Grant, a member of the Center for Great Lakes Literacy, a coalition of educators across the Great Lakes Basin. And we're featuring Ben Peterson of the School of Freshwater Sciences at UW-Milwaukee. Let's go ahead and get started. I'm going to close my video and close my audio. And if you are ready, go ahead, Ben. Thank you.
Okay, thanks, Ginny and Ann, for inviting me to this, and for all of you for attending. So today I'm going to be talking about my research on mercury, and kind of the take-home message of a lot of the work that I do is trying to understand how mercury becomes toxic. And not to give away too much of what I'll be talking about later, but essentially looking at the microbial transformations of this pollutant to understand why it has become such a kind of an important topic for research. So I'll spend the first little bit of the presentation just kind of talking about my background and kind of how I got into this research and kind of into the field of freshwater sciences more generally. I'll give an overview of mercury cycling, kind of a high level picture of what that looks like, and then we'll kind of briefly go through a couple of the research projects that I've been working on and that I've really been starting up here at UW-Milwaukee over the last year and a half. So first, background. So I grew up in Nakusa, Wisconsin, kind of right in the middle of the state, small paper mill town right on the Wisconsin River. And that has, like, I look at kind of where my career has gone and what I've ended up doing, and I can kind of see the seeds of that from growing up there. Spent a lot of time outside, a lot of time on the water fishing, camping, all those kinds of things. And I had been interested in science at like a really young age, but the kind of interests in science and the interest in the outdoors, I didn't really see a path forward to those kind of merging into one thing until one kind of later on. So I did my undergraduate work at the State University of New York at Geneseo. I was in the biochemistry program. I was looking at plant defenses and growing plants in a lab and measuring compounds that they were producing. I didn't really know what I wanted to do after I graduated. So I know a lot of you students are, kind of far away from that stage, but it really does take a while to kind of figure out, where you want to go. And sometimes even when you're all the way through college, you're not quite sure yet either. So I went and worked for the National Institute of Aging in the National Institute of Health. I was working in a neuroscience lab studying the brain and exercise and making mice run on treadmills and measuring how that affected their brain and things like that. And I really liked the science of it, but just being stuck inside and kind of doing the daily grunt work was it was just not what I wanted to be doing. Um, so I started thinking about graduate school and where I wanted to go and what I wanted to do. And I came across this program in environmental chemistry. And I started thinking, it's like chemistry outside, like that's interesting. That's not something that I had really considered before or thought about as an option, and started looking into that and ran into this study of limnology, which is the study of freshwater lakes, environmental microbiology, looking at just all of the organisms, the massive number of microorganisms that are present kind of on every surface everywhere and influence all these different things um, that we see. And I started seeing what people in the field were doing, going out and scuba diving, looking at the impacts of invasive mussels on lakes, sampling water from these large hydroelectric reservoirs, putting out buoys, monitoring lakes, and all these kinds of things. And I really got interested in that field, and it felt like something that could kind of merge these two interests um, that I had. And that's kind of how I ended up at the University of Wisconsin-Madison in the environmental chemistry program. I was fortunate enough that there was a project available there looking at kind of mercury and microorganisms and some of the things that I'm going to be talking to you about later. And I worked with Dr. Trina McMahon there, and it was just a really fantastic experience. I got to work on a lot of different lakes and a lot of different systems. Got to travel around the world to different conferences. Worked with a lot of great scientists, mentored a lot of great undergraduate students, and kind of all together just had a really good experience kind of learning about kind of mercury and how it works and how these microbes are interacting with it. So I kind of wanted to continue doing that. So I went and I did a postdoc out in California at the University of California, Davis, with Dr. Brett Poulin. Continued to be able to work in a lot of different ecosystems there. Spent a fair amount of time up in Alaska and these kind of remote areas that we would get into by helicopter and looking at the mercury that's in these kind of remote areas where the permafrost is melting, and in these kind of alpine rivers up there. Got to do some work down in the Everglades. I got stung by a scorpion down in the Everglades, which was not very fun, but the rest of the trip was great, and just kind of generally got to kind of go all over the place and investigate this in a bunch of different places. So then I've had the opportunity to end up where I am now. So a year and a half ago in the fall of 2024, I moved to the University of Wisconsin Milwaukee in the School of Freshwater Sciences, where I'm an assistant professor. And this is a really kind of special institution. I'll kind of give the School of Freshwater Sciences plug here. But we're the only school in the U.S. That's dedicated entirely to the study of fresh water, and it's really all aspects of water. So from the things that I'll be talking about today, like contaminants and microbiology, water chemistry, fisheries management, all that stuff, but also like water policy and economics and things like that. So for students, high school students, even middle school students, if you're interested in the things that I'm talking about today and or just kind of interested in water in general and kind of want to pursue a career in that, this is a really fantastic institution for that. So that's my background. I'll pivot now to doing more of a background on mercury cycling and to try to set up some of the projects that I've been working on. So mercury, oftentimes, even when doing presentations like this, I don't have to tell people too much about what mercury is because it's such a pervasive and a common contaminant. It is found in fish kind of all over the world. And we know that it has neurotoxic effects. So it can affect the brain function, especially in kind of young children and developing infants and babies. And so kind of one of the manifestations of this in popular culture is the Mad Hatter from Alice in Wonderland. So that um, the hatters, who used to make these felt hats, 100, 200 years ago, mercury was involved in the process of making hats. So the people who did this were often exposed to really high concentrations of mercury for a really extended period of time. And it would often, cause this kind of sort of degenerative brain function over time. So that is kind of what that character is based on, is kind of this true fact that a lot of the people in that profession were being poisoned by mercury as a part of their work. And so that's a pretty extreme example of the kind of the effects that it can have. But even at lower kind of doses, it can have a pretty negative effect um, on people. And it's bad enough that in Wisconsin here, that every lake, there's just a blanket fish advisory consumption for every lake in the US. So some are in Wisconsin. So some lakes have higher fish consumption advisories. So this is like the sign that you see kind of on the right side of the screen, that blue sign. Almost any lake that you go to in Wisconsin will have a sign like this posted on it telling you kind of what the limitations are on eating fish from that lake. And it's mercury is almost always kind of a part of that equation. So this is really I mean, this is a factor limiting how much of our natural resources that we can use right here in the state. And it's beyond just Wisconsin too. There's an international treaty. It's the first international treaty to kind of limit an element, but it's called the Minamata Convention, named after the city in Japan where kind of mercury, the first kind of mass poisoning event by mercury was found. But so that's a treaty that's designed to kind of limit the overall emissions of mercury across the globe among all the countries that have signed this agreement. Um, and for reasons that I'll talk about on the next slide, mercury is found in every corner of the globe. There's not, I mean, if you go to the most remote places in the Arctic or the Antarctic, you're going to find mercury in the organisms there. And there is some kind of naturally occurring mercury, but most of it is stuff that we put there. So since the Industrial Revolution, since we have started really burning a lot of fossil fuels and kind of revolutionizing a lot of production and things like that, we've increased the amount of mercury that's in the environment by about 3 or 4 fold. So really have accounted for a lot of the mercury that's actively moving through our waters, moving through our air, that's landing on the soils, things like that. And about 80% of the total mercury that's been emitted by people has been through coal, the burning of coal in these coal-fired power plants. So coal is heavily enriched in mercury. When it gets burned, like you see in this picture here, that those clouds, I mean, they contain a lot of things, but mercury is among one of them. And because of kind of the chemistry of it, which I won't get into too much, it can stay in the atmosphere for a long time, for up to a year. So you think about, these plumes of smoke going up into the atmosphere, kind of getting mixed in with the air that's circulating around the globe. And over the course of the year, it can travel pretty far. And so that's how it makes it to, the Arctic, the Antarctic. So the mercury that's being released, in Wisconsin, some of it may fall in Wisconsin, some of it's going to fall in Ohio, some of it's going to land in Florida, some of it's going to make its way across the Atlantic Ocean and land in Europe or Africa. And some of it'll, make its way up to the Arctic. And similarly, the mercury that's being deposited in Wisconsin, here in the and in other parts of the U.S. is not necessarily coming from here. It could be coming from really anywhere in the world. So it's really a global source. There are more local sources of it too. Industrial. A big one actually in Wisconsin is pulp mills. So paper mills can often be a source of mercury, especially historically, not so much anymore. But gold mining is a big source of it. So mercury binds very strongly to gold. It's used, miners mix mercury in with gold in the sediment or the kind of the mud that they're pulling out of the rivers. It pulls the gold out, and then they can take that ball of mercury and gold and burn off the mercury, and then the gold kind of remains, and they can, do with that what they want, as it's just pure gold. But all that mercury that they're burning off kind of is in these coming off in the smoke in the fires that they're standing around. So this is common in tends to be more common in poorer parts of the world. It's obviously an important economic engine for these people, but the health effects of it on them can be pretty negative. So kind of both these global sources as well as these local sources. But one thing that's interesting about mercury, and one of the things that makes it so fascinating to study, is that if you look at the concentrations in the environment, so even in all of these lakes in Wisconsin that have um, these fish consumption advisories, you would think, oh, there's a lot of mercury in the fish. There must be a lot of mercury in the water. And when I go to a field site to study mercury, I get people who live in the area asking like, oh, is the water safe to swim in? Like if you're here studying the mercury problem, is it dangerous to swim in the water? And the answer is usually no, that swimming in these waters is very safe. And that's because the lake, the concentrations of the mercury in the lake is really quite low. So we're talking about 1 part per trillion. And that means for every trillion parts of water and everything else that's in the lake, there's only 1 part of it that is mercury. And that's very hard to visualize. But a comparison is if you think about 35 Olympic-sized swimming pools, you think about kind of the massive size of that. So that picture there, that's standard Olympic-sized pool. If you take 35 of those and you dissolve a single grain of sugar across those 35 Olympic-sized swimming pools, that's 1 part per trillion. That's how much mercury is in these lakes that we are considering to be pretty contaminated. Um, Pretty small amount, right? So you can imagine that there's something else that has to happen for this to be a problem. It also means that it's really hard to measure this. If you're interested in looking at the mercury in a lake, and you have to get out this tiny amount of mass from this kind of large volume, you need super specialized equipment for it. And we're fortunate that some of the best kind of mercury instrumentation and the best mercury analyses analysts, scientists are right here in Wisconsin over at the Mercury Research Lab in Madison. That's part of the U.S. Geological Survey. So the problem with mercury is not with the concentrations that are in the water, but the concentrations that are in the fish. So when we look at these kind of large predator fish that people like to eat, like salmon or lake trout, if we're thinking about Lake Michigan, muskie or pike in inland waters. If you get out to the ocean, sharks, tuna, even mammals like dolphins tend to have really high concentrations of mercury, and it can be 40 million times more concentrated than it is in the water. And this is the part where it starts to become a health risk and become dangerous for people um, to consume. So the question is, how do you go from, 1 part per trillion uh, in the water to 40 times more concentrated in the fish? Um, and the answer is through these two processes. It's called bioaccumulation and biomagnification. So they're related but slightly different processes. So bioaccumulation is an increase in the concentration as the organism grows. So if the fish starts out tiny little fish, it's only eaten a little bit of food in its life, the mercury in its body is really, really low. But as it gets older and it continues to eat more food and swim around in the water, it accumulates mercury over time. And the mercury stays in the body. And while, the fish is, excreting other things, the mercury just stays. And so even as the fish gets bigger and bigger, it's accumulating mercury faster than it's growing. So the concentration in the fish is getting higher and higher. And then biomagnification is kind of moving up food webs. So if you look at some insects that are like some zooplankton or in benthic invertebrates that are crawling around at the bottom of the lake, these tiny little invertebrates, the mercury is really low. But then you look at, say, the perch that are eating them. Um, the mercury concentrations in those would be a little bit higher. And then you're looking at the muskie or the pike that are eating the perch, the concentrations are going to be higher there. And then you take, like a bird that's eating the muskie, it kind of gets higher and higher as you move up that food web. So that process is called biomagnification. Um, and both of these processes only occur with a very specific form of mercury called methylmercury. And methylmercury is not the form of mercury that's released to the environment. So when you burn coal, you're not releasing methylmercury. You're releasing this form called inorganic mercury. Um, with the gold mining, you're not releasing methylmercury. Any medical waste, any one of these sources of mercury, it's never methyl. So how do you get methylmercury in the environment that's causing these processes? Well, the answer is bacteria. So in the environment, there's naturally occurring bacteria that take that inorganic mercury that's released to the environment by human sources. They take it up into their cells, they convert it to methyl mercury, and then they spit it back out. And this then kind of triggers this ability for the methyl mercury to make its way into the food web. It gets into um, the algae and plants and things like that. And then it slowly works its way up the food web, getting to higher and higher concentrations until you're at kind of the top of this food web in these large fish, where the mercury is at toxic levels, essentially, and it can start to impair the development of the fish, and it's problematic for any humans who are then eating it. So these bacteria are a really key component of this mercury cycle. I like to think of them as kind of the gatekeepers to fish contamination, because if these bacteria didn't exist, we really wouldn't worry that much about mercury as a pollutant. It just wouldn't really be an issue at all. So the idea is that if we can understand these microbes, where they live, kind of why they're doing what they're doing, then we can start to understand mercury cycling and mercury pollution kind of more generally. Uh, so who are these organisms? They're like I said, they're naturally occurring. It's not something that we've released into the environment. They're found there naturally. They're often associated with environments that don't have any oxygen. So it's not, they're not. It was originally thought that they were not living like in the surface water of lakes and things like that, but rather that they're found kind of deep down in kind of the sediments, the mucky parts of the lake that the oxygen can't get um, down to. But we still, after decades of studying these organisms, we still don't really know a whole lot about these organisms. We know now that they have a gene. There's a very specific gene that they carry called HGCAB. Um, and that gene It's the one thing that will produce methylmercury. If they have the gene, they produce methylmercury. If they don't have it, they don't. Um, and from what we can tell, it's the only kind of pathway that kind of exists in the environment. And we know that the gene itself is very, very old. It has been present in organisms for billions of years. It's likely that it kind of existed in kind of we call it the last universal common ancestor. So the organism that existed before bacteria and humans and animals and all that. But when it was all kind of one like tiny organism billions of years ago, it had this gene for some reason. But the problem is we don't know why they have it. It doesn't seem to help the organism survive in the presence of mercury. We're not even sure that it's intended to produce methylmercury. It might be an accident. But there's these organisms, they're present kind of all over the environment. Um, they can produce this toxic compound, and we don't know why. So that kind of comes to my research goal. So my kind of whole PhD work, really for the last 10 years, and the research that I'm setting up now here at Milwaukee, which I'll cover just very briefly in the next couple of slides, is all focused on understanding these bacteria and using these bacteria as a way to kind of understand how mercury and other contaminants to a certain extent, but mostly mercury, is how do these behave in the environment? So where do these organisms live? What do they eat? They don't eat in the same way that we do, obviously. And then why? Why do they produce methylmercury? What's the objective for them? And the goal is that if we can understand this better, then maybe we can manage it a little better and try to kind of tip the scales in one direction or another in some of these ecosystems to try to limit how much methylmercury is being produced. And if nothing else, at least just kind of predict when and where that's going to be produced, and develop guidelines that would kind of allow us to avoid those fish that are kind of living in those areas. So again, just briefly, I just wanted to highlight a couple of the projects that my group is working on to try to answer some of these questions. The first is looking at the production of methylmercury in Lake Michigan. So I mentioned that these organisms, that these bacteria are thought to only be present in kind of in the muck and in the sediments, away from kind of the surface water where a lot of the fish are living. But we have some evidence that in Lake Michigan and in the other Great Lakes is that the methylmercury production is actually happening in the water itself, and there's tons of oxygen present in there. I mean, the fish all need it um, to survive, so there is tons of oxygen around. But what we suspect that's happening is that the bacteria are living on these tiny particles that are suspended in the water, and we're getting kind of these very localized conditions that are allowing them to have these kind of different groups of bacteria living kind of on these specific particles than are just swimming around in the water. So we've been working on this for the last year or so. We needed some specialized sampling equipment. So you can see this large tubing down here is essentially designed to just collect a ton of water all at once, to kind of to get enough of these small particles that we can start to look for these organisms. And essentially, kind of our research strategy behind this is that we're looking for these regions where there's a lot of the methylmercury being produced. And once we identify those, then we'll go in and we'll start kind of looking for the organisms that could produce it. And then kind of the third step is analyzing these organisms. And this is you'll see this kind of as a common theme through my work, is that we analyze these organisms to try to understand what environmental conditions impact these microbes, and use that as a way to understand kind of how and why the methylmercury is being produced in these different areas. So that's one of the projects. We're also working on a project looking at the intersection between mercury and the invasive quagga mussels. So if you're not familiar with the quagga mussels in Lake Michigan, They're these invasive species. Dreissena bergensis is the Latin name, but they invaded the Great Lakes and Lake Michigan specifically about 25 years ago, and they've completely reshaped kind of the food web and kind of destroyed the habitat for a lot of different organisms. And yeah, they've just really changed how it works. And the reason that they can do that is because they've been massively successful. So there's the picture of the quagga mussel up there on the screen. That the scale of that thing is about the size of a fingernail. The largest ones are about the size of a fingernail. The smallest ones are pretty small. So they're small organisms, but it's estimated that at their peak there were about 950 trillion of them in Lake Michigan. So you can imagine just kind of the massive numbers of them just blanketing the bottom of Lake Michigan. And they can filter the whole volume of the lake. They're filter feeders. They pass water through trying to eat the algae that are in the water, and they can filter the whole volume of the lake in about a week. Um, so th because they are filtering all the water out, they're filtering all the algae out of the water. The food web that of organisms that used to live up in the water and feed on the algae in the water are no longer there. They're all getting filtered out, and all those nutrients are getting moved down to the bottom or the benthic zone of that. So the quagga mussels are down here on the benthos, and they're bringing all those nutrients down here. So now the food web is kind of moving down there, and for mercury, that's potentially a problem because there tends to be more mercury in the sediments as opposed to the water column. So we're concerned that this is increasing the amount of mercury that's making its way into the food web. So kind of our initial questions for that are looking at is there more methylmercury in the benthic based food webs versus the whatever's left of the pelagic food webs, essentially in Lake Michigan. Where is methylmercury being produced? Where are the bacteria that are producing it? And then again, analyzing those organisms, trying to understand what factors in the environment are really driving their activity. Um, oh, and then I'll just mention that I have a graduate student. So the picture on the top right there is of Nicole Price. She's a master's student in my lab who's been working on this project. And then the last part of it is not based in the field. So sometimes some of the questions, even if you want to answer a question about what's happening in the field, you need to kind of reduce the complexity of it. You need to do some lab work first to try to get at some of those questions. So we're growing some of these methylmercury producing organisms in the lab under these highly controlled conditions, and we're trying to answer some of these questions that I've been mentioning throughout the presentation of essentially why are they doing this? So does the presence of the gene help them tolerate being exposed to mercury? It doesn't seem that it does, but we're kind of still looking into that. Do they produce more methylmercury if they're stressed, if if there's not enough food for them to eat, or if there's some other toxin? Um, there seems to be some sort of interaction with arsenic that we don't really understand yet, but that's another thing that we're doing. But with all this lab work that we're trying to do, we're always trying to kind of relate it back to the environment. So if we can understand why they're producing it, how does that inform, where they live, kind of in the environment and what they're doing, and how do we tie that back? Essentially, at the end of the day, what we're really interested in is how does it tie back to the mercury getting into the food web? How does it impact kind of fish concentrations of mercury? So just to kind of bring it back to this overall goal, again, all of these different projects that are kind of going in different directions are really kind of focused on that. And it seems sometimes like, studying the bacteria and getting into the nuances of what they eat and where they live and kind of what conditions they like to be under and kind of why they do it in the first place, seems far removed from, a fish consumption advisory. But because it sits at that kind of critical point, of converting the inorganic mercury into the methyl mercury, all of these types of questions are really important to address, and yeah, that's what we've been working on. So with that, I'd be happy to take any questions. I have my contact information up there. Always happy to get emails from people, so feel free to reach out. Um, yeah, and thanks again for coming.
Yeah, thank you, Ben, for sharing your work and the goals of your work and some of the students that are involved as master's students and how you got to where you are today. We do have a question and answer link, I guess, at the bottom there. So please go ahead and start typing some questions in if you haven't already. How we have managed this in the past, and we can decide if this is how we want to manage it again today, is that in order to have a smooth recording, we do need to have the audio of both the question and the answer. So in the past, some of the presenters have asked that I read the questions and they answer, and others have preferred that they read the questions and then go ahead and answer them. So first thing we'll do is, Ben, if you can let us know which method you prefer, and we'll facilitate it that way.
I don't mind reading them. That's okay.
Okay, so we'll allow you to read the question and then go ahead and answer it. And you can answer them in any order that you want. You don't necessarily have to answer them how they came in typed. If you see things that seem to be grouped together in your mind, please feel free to answer it that way. And for the audience, please continue to write your questions in that Q&A area.
Hmm.
So we'll let you start whenever you're ready, Ben.
Yeah,
Thanks.
for sure. Um, so let's see. So Becca asks, is focused on predatory food webs. Is there much knowledge on methylmercury biomagnification mechanisms in aquatic plant life? There is work on that. Yes. I'm not as familiar with kind of what that looks like with um with the aquatic food webs. Typically you're most worried about mercury pollution in these kind of high trophic levels. So fish that eat other fish, because of that biomagnification process, and you get that kind of increase in mercury concentration with each step. So in phytoplankton, it's low, algae, it's low. The zooplankton is a little bit higher, kind of the prey fish that are eating the zooplankton and the insects are a little bit higher, but then it starts to get really high when you're eating, especially if you have like a fish that eats a fish that eats a fish. Um, so with plants, it's typically not as much of a problem. There are some plants that accumulate mercury at high concentrations, and I know that rice is one of them. So there's a lot of work, um, especially being done in China. There's a lot of Chinese groups that are doing a lot of really great work on mercury concentrations in rice paddies, because the rice paddies tend to facilitate conditions for methylmercury formation. And then they're also efficient at taking it up. So it can be a problem in some areas. But in terms of other plants, I'm not too sure kind of how much of an issue it is. Uh, Someone else asked, is it safe to eat fish if there is mercury in them? It depends on how much mercury there is and how much of the fish that you're eating. And it also depends on who you are and kind of where you are in your developmental stage. So the people that are most susceptible to mercury poisoning are young kids, essentially the kids that are developing. So it's recommended that women who are pregnant or expecting to become pregnant try to limit their mercury intake, because the mercury can cross the placental barrier and impact any um, uh, developing fetuses. And then for children as well, when you're kind of in that period of rapid brain growth, that's when mercury can kind of have the most adverse conditions. It can impact other people, but for like older adults, it tends to not be as much of a problem. And your body does get rid of it after a while. So you can eat, even for people who are in these higher risk categories, there's some recommendations of like how many fish you can eat, how many of certain types of fish you can eat in a week, or things like that. So it really depends on who you are, what the fish is, and kind of where the fish is living. Um, Someone else asked, how would you know if bacteria is stressed? That's a great question. There's a lot of different ways that bacteria can be stressed. So they can be stressed by not having enough food. Uh, they can be stressed by kind of getting um, dried out. So if they live in kind of along the edges of waters and the water level drops, things will dry out, and they'll become stressed. If they're exposed to too much light, other contaminants can cause them to be stressed. And these different stressors kind of elicit different responses in the bacteria. Oftentimes, what we do is just, we mess with them in some ways. We take away their food, or we expose them to light, or add some other contaminants that we know are dangerous for them. But there are a lot of people who's kind of specifically studied the stress responses, and there's a whole bunch of different kind of genes that can get turned on in response to these different stresses. Titus asks, are there ways to remove mercury from aquatic environments? That is a really good question. At this point, there is not a way to do it at scale. I mean, you can, yeah, no, essentially no. I mean, at the scale that you would need to do it in these systems, there is not a way to do it. The good news is, I'll say the good news about mercury is that mercury emissions globally in the US specifically, but or in the US especially, but even across the globe, mercury emissions have declined over the last 40 years. And it has a lot to do with kind of air quality regulations, not even for mercury, but for other, like for the SOx and NOx they're called, but kind of sulfur and nitrogen limitations on emissions. The mechanisms that people use to control those emissions also can be somewhat effective for mercury. So even in the coal, like the continued coal fire power plants that have been operating over the last 40 years have been releasing less and less of this mercury because they've become more efficient at removing it before it um, gets out. And we've seen a decline in water concentrations of mercury that kind of parallel that. There's still a lot of mercury in the fish because of this kind of importance of the methylmercury component and other factors that may be increasing the amount of methylmercury in the environment. But it seems that the overall mercury um, kind of burden in the environment is decreasing, at least for now. Um, so would be a more effective way going forward. I would say is to kind of control the point sources and then kind of just wait for it to get out of the systems. If money wasn't a factor, what questions would you really love to answer about marketing? Oh, that's a nice question to think about. Um, I mean, the question that has really bugged me since the start, since I um, started my PhD. So I started my PhD in 2015, and the HGCA gene had been discovered 2 years prior to that. Before that, we didn't even know what gene was responsible for it. But that whole time, the thing that I've really wanted to know is why, what is it for? Why do they have it? Like, what benefit um, does it serve to them? Because every time we go into a system and we, spend all this time and money trying to figure out, when and where um, these organisms are living, we can kind of pull out some patterns, but um, if we look at kind of the distribution of these organisms along different, gradients, different like nutrient gradients. In one system, it'll be one way, like as you get increasing, like sulfate is a common one. As you get increasing sulfate, you get more of these organisms. In other environments, as you get increasing sulfate, you get less of these organisms. And it's it's probably not impossible, but it's nearly impossible to say why that's the case without knowing why the organisms do it. Because we don't know what they're we don't know what they're responding to. Um, so that, and I think that's like a really big question, and probably I wouldn't say it would take an unlimited source of money, but it'll take a lot and it'll take a long time. Um, so if we had unlimited funds and we could just put like 50 people on it trying to figure it out, that's probably what I would do.
For now, though, that's the questions in the question and answer space. Please do feel free to continue to write more if you'd like. And I have a question for you. If you'd be so kind to, if you're able to back up one slide, I think it was just one. Maybe two. There is yes, there. I'm curious as to what the test tubes in the image are indicating and why one is purple and the other is pink. What were you testing for and what does that color change indicate?
Hmm. Yeah, so this was the picture here is of my an undergraduate student, Grant, who actually just graduated last semester, and he was kind of doing some of the preliminary work in the lab just to get the organisms kind of up and kind of growing and happy and surviving and producing methylmercury in the lab. So what he is doing here was testing out some growth conditions for them. And in this case, essentially the purple one was um, uh, he was using some indicators to kind of test if the growth conditions were sufficient for the organisms to survive. And in the purple one they were not, and in the pink one they were. So this was indicating a kind of a successful growth of one of these organisms, which he worked long and hard to get to that point. So.
Yeah, thank you.
Mhm.
Um, in we have another question that came in. Someone has asked, what did it feel like to be stung by a scorpion?
It felt like
Obviously
a
memorable.
Yeah, it felt like a bee sting, and I'm allergic to bees. So that's why it was very concerning. I was relieved to find out that it was a scorpion, and not a bee, because I got stung on my neck a couple of times. Um, but yeah, it's almost identical to a bee sting is the feeling. But apparently, if you're allergic to bees, you're not necessarily allergic to scorpions, which is good.
Yes, very, very fortunate. Another question is, how many different types of bacteria are you working with? And are there many of these types of bacteria that have this gene and are able to do this?
Yes, there are. Actually, I think I had a um, image just up. Um, so in the lab, we only work with, we have probably, I think we have 40 organisms that can produce methylmercury in the lab. We really only work with two of them, just to kind of reduce the complexity of it. Um, but in the kind of globally, there are like in the environment, there are massive numbers of them. And bacteria are diverse enough that we couldn't really put a number um, on it. There are I mean, probably 100 isolated organisms that can do it, but in our surveys of the environment, um, I mean, thousands of them have been identified, and that's every time you go into a new environment, there's hundreds more. And that's just kind of a function of how microbiology is, that the um, the numbers of species and even higher taxonomic levels is staggering. And this gene is present throughout a wide variety of them. And I mean, since it was, as I was mentioning, it was present in the last universal common ancestor, which means that kind of the origin of kind of life as we know it had this gene in it at some point. So any organism that doesn't have it lost it at some point along the way, kind of in its development. So it's very widespread. And yeah, there's a here, let me see if I can switch my screen over to okay, so this is a figure from a paper that one of my fellow grad students when I was at Madison published, and it's a tree of life essentially. So all the different branches represent a group of organisms. Um, there's so I think it's just back to, I don't think the eukaryotes, so like humans and stuff aren't on here, but it's bacteria and archaea. And all of the different purple spots represent a group of bacteria that have been identified to have the gene. And I mean, this was from 5 years ago, so I'm sure there's many, many, many more of these dots. And within each of these dots, there's, dozens, if not hundreds of organisms with it. So yeah, very diverse kind of group.
Thank you for showing that extra figure. I think that's really interesting. Another kind of related question is how did you go about deciding to choose the two that you did that you're using prominently in your lab? Why those?
Hmm. Um, sorry, this is um, so one of them was the organism that it was originally discovered in. So there's a group from Oak Ridge National Laboratory who identified it. And that was in 2013. And when they first found it, they then looked in other organisms that we had in culture, and there were maybe about 50 or so total that had the gene that we had isolated and were growing in labs. And the one that they were working with is, I mean, the reason they found it in that one is it's a very easy organism to work with. Compared to the other ones, it grows relatively easily. It's still a little bit tricky because it can't tolerate any oxygen. But it's easier to work with than the other ones. And the second one is kind of a similar thing. It's different enough than the first one, so you kind of cover this kind of range of different types of microbes, but they're relatively easy to work with. Um, Mercury is very difficult to work with, and it's hard to analyze and all that stuff. So any part of it that you can make a little bit easier just makes the kind of overall process easier. But yeah, they're common kind of workhorse bacteria for that. So it'd be great to expand out to other ones because, something that might be true for one organism is not necessarily true for another, but um, just kind of funding and time constraints limit it for now.
Again, thank you so much for your work that you're doing and all of the insightful information that you and your colleagues are gathering. It's really incredible, all this different aspects that we still don't know very much about. Um, I don't have any other questions here, so I think I'll just take the last couple minutes of our time together to once again let folks know that the presentations are being conducted by the Center for Great Lakes Literacy in cooperation with Wisconsin Sea Grant today. We are serving as the host. If you are interested in a recording of this presentation, it will be available in the future. It does take us a while to make the transcriptions available, and we don't post the recordings until the transcriptions have been completed. So look for that on the Center for Great Lakes Literacy website, which is siegel.org. And then if you do have additional questions, you can reach out to either me or to Anne with them, and we will attempt to either contact Ben if we are not able to answer it ourselves, or hopefully just go ahead and give you an answer. We don't yet have another presentation scheduled for the spring, but we do anticipate doing that shortly. So if you're interested in future Students Ask Scientists presentations, again, watch the Center for Great Lakes Literacy website or continue to follow our listservs through the Wisconsin Sea Grants education pages. With that, I think we'll say thanks once again to Ben, and thanks to the educators who have made it possible for their students to attend with us today. And if you could launch that poll one last time. If you weren't able to join us right at the very beginning of the presentation, we did have a poll that helps us with our reporting requirements. We'd ask you to complete that, indicating what state you're from, what city, your zip code, and how many individuals you had with you today. If you've already completed that poll, thank you very much. And we look forward to seeing you again at a future Students Ask Scientists presentation.